Okay, there are 9 people here. Say hi. You're already live. Okay.
Right. I respect that. I respect that. Great from posting, or great from being active in any way, like you and
Okay.
such a good friend.
I just want to make sure like nobody kicks it, so I don't have to be like, excuse me. Kilan, did you put the live streaming link on your Facebook?
It's like his invitation's still waiting for me. I've accepted everyone.
I didn't notice it until you brought it up. That shows.
No, it's okay.
No, I like sat down in my chair.
Did you do that on purpose? Yes.
Such a baby.
I was like, you might as well make it a theme. Pink shades, breast cancer.
No point in whispering.
See, stuff like that. Anyway.
Right.
Okay, so let's wait.
He's on his way. He's on his way.
But let's get started. So it's a great pleasure to welcome everybody, Kilan's family foremost, who came all the way from California to be at her defense. And so today is a big day. It's a great pleasure to introduce Kilan. Kilan is originally from California, but moved to Miami in 2012, where she joined the PhD program here at the University of Miami. Then Kilan joined my laboratory in May 2015. And since then, things have gone really well. So I should mention she was actually the first one to join my lab after my laboratory moved to the Department of Surgery here at UM. And she was also instrumental in getting the lab up and running as the first person. And in particular, Kilan has been exploring the role of a completely novel transcriptional regulator in the oncogenic Wnt signaling pathway. Why is this important? This gene and also the Wnt signaling pathway are preferentially overexpressed in a very aggressive form of breast cancer called triple-negative breast cancer. This type of breast cancer is more prevalent in African American and young women, and we currently don't have any specific treatments for it, but this factor actually looks like that could be a very good potential target that may have actually very little side effects in patients, and maybe Kilan will allude to this. She, as I said, the work has been very successful. She just published a first author paper. I also actually presented this work. I just got back from San Antonio, Texas, where I presented her work at the San Antonio Breast Cancer Symposium, and it was very well received. A lot of people came to see the poster, so kudos to you. And she also actually participated in other projects in the laboratory that examined actually the role of this protein more in human breast cancer, and so there are a couple of more papers coming out in the near future. Apart from her lab work, she also actually has gotten a few distinguished awards. So she presented a 3-minute thesis presentation here at UM and got first place in the People's Choice Award and the second place overall. So that's also awesome. And another thing worth mentioning is she was also invited to give a talk at Yale University, so one of the Ivy League schools, at the meeting of the Ashad-Bishop conference. So they selected her to present an oral presentation, so which is not a small feat. And so without further ado, I'll hand over to Kilan, and she's going to present her interesting work, and I hope everybody can actually follow, even not in science, but I'm sure she'll do it for sure.
All right. Thank you, Dr. Riegel, for that introduction. My name is Kilan, as you guys know at this point. Thank you all for coming out today to hear about my work, elucidating the role of the transcription cofactor LBH in the etiology and progression of aggressive triple-negative breast cancer. This work was done under the guidance of my research mentor, Dr. Carolyn Riegel. So before we get too deep into things, I'm going to give you a general overview of what I'll be talking about today. So before we get too deep into breast cancer, we'll be talking generally about the breast anatomy, because the breast mammary gland that makes up the breast is a complex organ that leads to various types of breast cancer, including the subtype that we study in particular. Then we'll be talking about our gene of interest in the lab, limb, bud, and heart, and a few of its roles in cancer that we've found out over the course of this work. Breast cancer is the second leading cause of cancer death among women in the United States. In 2018 alone, it's estimated to there are estimated to be about 260,000 diagnoses and 40,000 fatalities. And what's interesting about breast cancer is that it's not just one disease, but it actually can be characterized into various clinical and molecular subtypes, and those have varying implications that we'll discuss later. But before we get too deep into that, we need to talk about the breast. In particular, we need to talk about the mammary gland, which is in the breast composed of ducts and lobules that all start from this one very interesting cell. So the way that the mammary gland is composed, we have a universal stem cell called a mammary adult stem cell. This stem cell was identified basically based on its capability to recapitulate an entire mammary gland upon transplantation, and that could happen from transplanting one cell. This mammary adult stem cell gives rise to bipotent progenitor cells that then give rise to lineage-restricted progenitor cells. And these are basal-myoepithelial or luminal in origin. And we'll talk a little bit about that in a second. But essentially, these progenitors then give rise to mature cells in the gland that organize to look like this. So this is a schematic of the mammary epithelium. And basically what you can see is that these luminal cells, represented here in blue, organize around the inside of these ducts and lobules, and the basal myoepithelial cells organize around the outside, separating this luminal cell layer from the basement membrane. And in these two different epithelial populations, there are stem cells that we talked about, but also these progenitor cells that are a little bit down on the hierarchy. And these different types of progenitors can actually give rise to different types of breast cancer. So essentially, there have been a few models for the cell of origin in breast cancer. It was originally thought that maybe one cell type, which would be over here, that mammary adult stem cell, gave rise to all the types of breast cancer. And then it was thought that potentially there were different cells of origin, and based on some genetic reprogramming, that a luminal cell might give rise to a luminal tumor, or a basal cell might give rise to a basal tumor. But it's actually just more complicated than that. Through a combination of differing cell of origin, but also genetic reprogramming, we actually get luminal, HER2 positive, basal-like tumors, and also these claudilometaplastic tumors. These luminal tumors have a pretty good prognosis. They express estrogen receptor and progesterone receptor, which are molecular markers, but also therapeutic targets. And we have pretty consistently effective drugs for those. We also have HER2 overexpressing tumors, also a molecular marker that serves as a therapeutic target. But these basal-like tumors are negative for all of those markers, and that's why we call them triple negative. Not only do these tumors lack those markers, but they represent about 15 to 20% of breast cancer cases. And their lack of those therapeutic targets leads to there actually being the highest fatality rate among breast cancer subtypes. As Dr. Riegel mentioned, triple-negative breast cancer disproportionately affects young women, given that breast cancer is usually a disease of postmenopausal women, and also African-American women for mechanisms we haven't completely figured out yet. It's also associated with being very aggressive and lacking these molecular markers that also serve as therapeutic targets. What you'll see here is a representation of some literature actually pretty uniformly agreeing that there are markers for basal triple-negative breast cancer. It's just that they tend to be cytokeratins, which are structural proteins, and not necessarily therapeutic targets. Therefore, there's an urgent need to identify reliable biomarkers in basal triple-negative breast cancer that may eventually serve as therapeutic targets as well. And we found one. So During her postdoc, my mentor, Dr. Briegel, identified this novel protein, limb, bud, and heart. And work in the lab basically found that it's a novel marker for basal triple negative breast cancer. Right here we have a normal breast, and LVH is restricted to that outer layer of basal and myoepithelial cells that I told you about earlier. In luminal and HER2 overexpressing tumors, there is virtually no LVH in the mammary epithelium. However, in the HER2 overexpressing tumors, there's some in the stroma, and that's important that LVH is expressed in tumors, in infiltrating cells in tumors. But where we see robust LVH expression is in that basal triple negative phenotype, or in that subtype. And that was also confirmed by looking at a panel of breast cancer cell lines. So you can see here that about half the cell lines are luminal, and a proportion of those are estrogen receptor positive. And here you see virtually no LVH mRNA expression. However, in these basal estrogen receptor negative cell lines, there is little to very high LVH expression. And this is actually a normal mammary epithelial cell line that is basal in phenotype. So it's also, there's just a correlation between LVH positivity and ER negativity, and also these very aggressive breast cancer cell lines that happen to be enriched for stem-like characteristics. We'll get to that. So what is LVH? LVH is a transcription cofactor that was given its name for its unique expression pattern in the developing limb, bud, and heart. Now, it's worth noting that LVH is not only expressed in the developing limb, bud, and heart. It's expressed in the peripheral nervous system, in the spleen, in the kidney, in the bones. However, it got its name, and it does have a very restricted, very unique spatiotemporal expression pattern in these organs. LVH is a direct target of two signaling pathways, the Wnt-beta-catenin pathway and the TGF-beta-SMAD signaling pathway, that have both consistently been demonstrated to have crucial roles in breast cancer and other types of cancers. And LVH itself is overexpressed in about 45% of triple negative breast cancers. What's interesting about LVH, and what you don't typically necessarily find, is that genetic ablation of LVH does not impair embryogenesis, which means in mice you get fully formed pups, so you can have a successful pregnancy. Those pups grow to be healthy adults, and then they actually have normal adult organ function. And recently there have been some defects characterized, but nothing that is lethal or impairs adult life significantly. Work from the lab, there's been extensive work from the lab characterizing the role of LVH in that mammary adult stem cell that I was telling you guys about. So using mammary epithelial cells for mice combined with cell surface markers and fluorescent antibodies, we're able to actually segregate that luminal population from the basal population on the basis of positivity for these markers. And there are various markers that can be used. This is just one agreed on, well-characterized set. What we found in the absence of LVH was that there was a decrease in the amount of basal cells and an increase in the amount of luminal cells. And so that led us to believe that LVH was promoting this basal lineage. And there's also a functional role. So when these cells were actually sorted, which we're able to do by this same kind of FACS analysis, and it's just a different end result, unsorted, so whole mammary epithelial cell populations demonstrated a decreased capability of forming mammospheres. Mammospheres are an assay that we use that basically assess stem cell capability. And so what we did is plated and found that there was decreased mammosphere forming capability, but when these luminal and basal populations were segregated, we actually found that the decrease was due to an effect in the basal proportion of cells. And that is where LVH is predominantly expressed, so that's what we would expect. Also, what was really interesting in these LVH knockout mice, these are mammary glands. So this is the way that structure looks, that inner layer of luminal cells and the outer layer of basal cells. So you can see that in normal circumstances, in these LVH competent glands, There's two very thin layers of cells, and that there's select positivity for estrogen receptor alpha and progesterone receptor. And both of these receptors and their ligands are crucial for mammary gland development and also play roles in breast cancer. When LBH is lost, there's a thickening of this luminal layer, and that's evidenced by an increase in the proportion of estrogen receptor positive cells, and also its target gene progesterone receptor. Now, that is of particular interest. So if a cancer biologist looks at that, it's of particular interest because, like I said, we have treatments for hormone receptor positive cancers. It's the triple negative cancers that are really aggressive and hard to treat. And so that was an exciting finding. There's another stage where these studies were done during puberty. And this is a schematic to show you kind of where puberty sits in the postnatal mammary gland development scheme of things. So from birth to puberty, there is an outgrowth that's tightly regulated and hormone responsive during puberty, and this is a fat pad. So basically, this mammary gland is growing out, and these highly proliferative terminal end bud structures are where the stem cells localize. So these are hyperproliferative structures that then lead to the invasion of the gland through the fat pad. And when these mice mature to adulthood, they're basically dormant until pregnancy. In preparation for lactation, there's another stem cell driven rapid expansion of the mammary tissue. And that stage, pregnancy, is also where LBH is overexpressed. We actually have demonstrated that similar to puberty, there is an effect of the loss of LBH on that expansion during pregnancy as well. So this all informs the conclusion that we made that LBH is a crucial stem cell regulator. But how is it doing this? So we took these HC11 cells that are it's a mammary epithelial cell line that actually retains its luminal characteristics. And that's important to note because some of the immortalized mammary epithelial cell lines become basal through the transformation process. But this one is luminal, allowing us to study what effect LBH is having on luminal and basal lineage specification. So basically, I'm going to direct your attention here first, because what you can see is that these cells are vector controls, and this is an LBH overexpression construct, and you can see a drastic upregulation of LBH. This gene, delta NP63, which is also a stem cell regulating gene and has some very crucial roles in development, but also cancer. We knocked down this gene in these cells and basically found that while, okay, so you have to bear with me because this is vector and LBH overexpressing, but then on each graph we also have a control, scrambled control, and also this gene is being silenced, okay? So what we had was basically delta NP63 is upregulated when LBH is overexpressed, and then we had confirmation that that upregulation was abrogated when we knocked down delta NP63. Cool, that part worked, the model works. So then these HC11 cells that overexpress LBH, we were able to demonstrate that P63 was actually still upregulated in this cell line. However, there was a decrease in the amount of spheres that formed when you knocked down delta NP63, indicating that basically delta NP63 was functioning downstream of LBH in increasing the sphere forming capability of these cells. We also looked at estrogen receptor alpha. And so estrogen receptor actually is repressed when LBH is overexpressed. There's an inverse relationship there. But that effect was abolished by the knockdown of delta NP63. This all indicated what I'll demonstrate here, which is that this is the model that the previous work established. LBH promotes the basal lineage and also the renewal of mammary adult stem cells via induction of delta NP63 and repression of luminal estrogen receptor alpha and luminal differentiation. In the context of LBH deficiency, not only is the self-renewal capacity of mammary adult stem cells reduced, and that we demonstrated with those mammosphere assays that I showed you, but also that delta NP63 is no longer upregulated, and then also that there's an increase in luminal differentiation and an increase in estrogen receptor alpha. Okay, so given that there was this defined role of LDH in normal stem cells, we wanted to see whether the expression of LDH was also correlating to some role for LDH in cancer stem cell regulation. And so, in order to find out whether that was the case, the first thing that we wanted to know is LDH upregulated in cancer stem cell populations? That's a good indication that it's probably doing something. And then we wanted to know whether LDH was actually driving stem cell activity past just being expressed. And so, basically that's important because if LDH is upregulated in these cancer stem cells, it may represent a very interesting treatment opportunity because these cancer stem cells have actually been identified as the underliers of treatment resistance, but also metastasis, which leads to the spread of cancer throughout the body and ultimately proves fatal. So, we took 2 of the breast cancer cell lines that I showed you earlier. The MCF7 breast cancer cell line is luminal. It is still tumorigenic. It's just a different type of tumorigenic. And we also took the BT549 cell line, which is a basal cell line. And they are low, they're negative and low for LDH respectively. So we took the, on the basis of CD44 and CD24 positivity, we took a highly differentiated luminal population from the MCF7 cell line, and we sorted that out. And then we also sorted out a CD44 positive, CD24 negative cancer stem cell population from the BT549 cell line, and also a more differentiated, but still progenitor-like CD44 positive, CD24 positive population. We did a real-time PCR, a qPCR analysis of LDH mRNA and found that LDH was upregulated significantly in the cancer stem cell subpopulation, and to a lesser extent in those progenitor-like cells. And so then we took these cells and we cultured them in tumor sphere cultures. So that same mammosphere assay that I was telling you guys about, when you do it with a tumorigenic cell line or with cells from a primary tumor, it's a tumor sphere culture, but essentially the same thing, way to assay for stem cell activity. So we did tumor sphere cultures of these cell lines and found that LDH was upregulated in both cell lines when these populations, or these cancer stem cell populations, were enriched through tumor sphere culture. And they weren't, LDH wasn't upregulated alone. LDH was upregulated in concert with other stem cell regulating genes, such as LGR5 and NANOG, and what you'll also see here is that delta NQ63, that LDH regulated gene that I told you about, is also upregulated, indicating that there may be a similar mechanism between normal development and cancer. So then we took cell lines that had already been made in the lab. These are the same BT549 cells that I was telling you about, but these have been, there's an LDH overexpression construct that's been introduced. So these cells overexpress LDH past the endogenously low levels. And when we used FACS analysis for those same stem cell markers that I showed you in the previous slide, we were able to demonstrate a small but statistically significant increase in the proportion of these cancer stem cells. We took another cell line, highly aggressive LDH high MDA and B231 cells, and we knocked down LDH, so that's opposite. And we found that there was a significant decrease in the CD44 positive, CD24 low metastasis-initiated cell population. And we used that criteria that had been established by another group here at the University of Miami. And they characterized that while both populations do have cancer stem cell activity, it's really the low population that underlies metastasis. Earlier, I showed you that basal triple-negative breast cancers can actually arise from luminal progenitor cells, right? And so the mechanisms by which this happens, this luminal to basal conversion, is an area of study that's really exciting and of particular interest to our group, given our demonstrated function for LDH. And so we took MCF7 vector and MCF7 LDH overexpressing cells. They were injected into mice, tumors formed. Then they were embedded into paraffin, and I did some histological analyses of these tumors and found that this luminal keratin, keratin 8, was downregulated, and that there was what was of particular interest is that there was an emergence of these keratin 5 positive cells in a largely luminal cell line, and keratin 5 is basal. So it was indicating, that was our first evidence that LBH overexpression was sufficient to induce a partial luminal to basal conversion in a breast cancer cell line. So then we did an immunofluorescent analysis, and here you can see the nuclei of cells in blue, and then the proteins are here indicated by which color they're represented as. So, like I said, MCM7 vector tumors are largely luminal and keratin 8 positive. However, when LBH is overexpressed, there's the emergence of, okay, we're not coming. There's the emergence of these keratin 5 positive basal cells. And what's interesting is that we were able to identify keratin 8 positive, keratin 5 double positive MCF7 cells. So it's not as if these are completely new cell type. That was really exciting evidence of this luminal to basal conversion. I probably did not mention that the MCF7 cell line is estrogen receptor positive. Outside of being luminal, it also is hormone dependent for its growth. And so basically what I have here is these vector and LBH overexpressing cell lines, and this is a representation of their cell cycle profile, so the way that they're growing and dividing. What you can see is that in these samples right here, that's the normal growth profile in full cell culture media. However, when the vector cells were cultured in charcoal strip serum, which gets rid of, which basically retains salts and amino acids that are necessary for cell growth, but gets rid of small molecules like hormone receptors and other fatty compounds. Basically, the cells go into growth arrest, where a large proportion of the cells actually stay in G0, G1, which is a pre-mitotic phase, and a smaller proportion of the cells are actually synthesizing DNA and then progressing to mitosis. However, when we overexpress LBH, the cell cycle profile, if you compare it here, looks very similar to normal growth conditions. So what does that mean? That means that LBH overexpression is actually promoting hormone-independent growth, which is a characteristic of basal cells in the MCF7 luminal cell line. So that was really exciting as well, and that's something that we need to do further characterization of. So what can we conclude from this part one? We were able to find that LBH is upregulated in cancer stem cell populations and also tumor sphere cultures. That LBH overexpression, up or loss, down, significantly affects the cancer stem cell population. And that LBH overexpression in a luminal cell line induces a basal conversion in these MCF7 breast cancer cells. And a part of that is the promotion of hormone-independent growth. All right. So that led us to part two, which is the role of LBH in Wnt-driven tumorigenesis. And you may have a question, what's Wnt? No worries, I got you. So, in normal development, Wnt is a family of secreted ligands that affect various processes through the Wnt pathway during development. And basically, there's canonical beta-catenin independent signaling, and that's really the pathway that promotes adult stem cell maintenance and self-renewal. But it's also a function that can become oncogenic when it's dysregulated, as it is in breast cancer. So aberrant Wnt signaling is observed in triple-negative breast cancer and also correlates with poor outcome in breast cancer patients. And what's interesting is that overexpression in vivo, which means like in a live system, so in mice, results in mammary gland hyperplasia and subsequent carcinogenesis that resemble basal triple-negative breast tumors in humans. And these tumors are enriched for stem-like characteristics. This is a schematic of the Wnt pathway. So this entire protein complex is what we call a degradosome, And essentially, its normal function is to phosphorylate and ubiquitinate, ultimately degrade this transcription factor beta-catenin. However, when Wnt ligands, which are here, bind to these frizzled and LRP5/6 receptors, the degradosome gets sequestered to the nucleus, which means that beta-catenin is free and can translocate to the nucleus, associate with other transcription factors, and drive the transcription of Wnt target genes like LVH. So previous work from the lab had demonstrated that LVH was upregulated in 6 independent MMTV Wnt1 tumors, and there's more that are represented here, and positively correlates with basal keratin 5, whereas there's an inverse correlation with luminal keratin 8. And these MMTV Wnt1 transgenic tumors have a large basal proportion of an expanded basal population where LVH is predominantly expressed. So here you can see LVH expression in the wild type gland restricted to that layer, that outer layer of basal myoepithelial cells, a very similar expression pattern for basal keratin 5. However, you can see that there's robust LVH overexpression in these MMTV Wnt1 transgenic tumors. And these are just spontaneous tumors that form in mice after a period of Wnt1 overexpression. And so for our study, we needed Wnt, but we needed to be able to model loss of LVH. So we started breeding some mice. So we took these MMTV Wnt1 transgenic mice and we crossed them with a conditional LVH knockout that our lab actually developed. And what this conditional knockout does is downstream of keratin 14 Cre, which is epithelial specific, epithelial cells, remember, that basal luminal. In epithelial cells, this flox LVH results in a truncated non-functional LVH protein. And so what we get is 2 genotypes that we actually studied. So we have the Wnt1 keratin 14 Cre mice, and that's not really the part that we need to focus on. What we care about is LVH, wild type or negative. So the reason I say that is because obviously there's a lot going on here. It can get a little confusing. Both of our study groups do have overexpression of the Wnt transgene, hyperactivation of the Wnt pathway. What we're modulating is LVH expression. And this is just qPCR, mRNA analysis, and then also Western blot protein expression in these mammary glands to show that we basically were able to generate LVH knockout mice, and that worked. So our research question for this part was, will genetic ablation of LVH in these mammary epithelial cells reduce Wnt-induced mammary gland hyperplasia and subsequent oncogenesis? And there are various parameters that we looked at that I'll go through now. So previous work had demonstrated that and we recapitulate that here, that there is a developmental delay in LVH deficient mice. So this is similar to that schematic of postnatal mammary gland development that I showed you earlier. And basically you can see that at 6 weeks, there's a significant proportion of the fat pad that's already filled with mammary epithelium and stuccal tree. However, when LVH is lost, outgrowth doesn't go much past the lymph node, which we use as kind of a marker for outgrowth. And at 8 weeks, that delay is less significant, it's less drastic, but it is still there. And that's quantified both here and here. As you can see, even at 8 weeks, there's about 40% decrease in ductal outgrowth. That's the normal wild type condition. Since we confirmed that we could reproduce some of the previous data from the lab in this model, we then went and studied our transgenic condition. And what we found is that LVH loss stunted growth of these MMTV Wnt1 transgenic glands at 6 weeks, even more than Wnt1 transgene overexpression already stunted growth if you were to compare here to here. Now that 6 week delay, there was a delay at 6 weeks, and at 8 weeks this had resolved, and obviously at 10 weeks. However, what is consistent in these different age groups is that there was a decrease in the overall overall thickness of this invading ductal tree. And we wanted to know why that would be the case. So we went to a 12-week mouse where the mice are firmly adults. And that's also the age where I showed you the expanded proportion of basal cells on the facts plot earlier. And so basically here you can see that there's a wild type gland. I actually want to draw your attention here to the close-ups because you can see that those terminal end bud structures that I told you about are basically gone, and that's what happens when you get a mature gland. The idea is, okay, I'm full, I'm mature, until you want to have some babies, I'm dormant. However, in the context of wind hyperactivation, there's still actually a lot of growth at this stage, and you can see that as it's evidenced by this very thick ductal tree. When LBH is lost, we get that same decrease in ductal thickness and a visual purported decrease in the terminal end bud structures. We did an H&E stain. So H&E stain is a very simple histological stain, and basically what you'll see is that the purple stain is nucleic acid, so that'll represent like a cell nucleus, and then the pink is cytoplasm, so it stains proteins, generally cytoplasmic. And so it's just a way of visualizing the cells in a tissue or in a section. And so you can see that there was a marked decrease in how much mammary epithelial tissue there actually was with LBH loss. And that effect was mammary epithelial cell specific because when we looked at collagen deposits as assayed by this mass and trichrome stain, you can see that there's no decrease in the collagen, which is represented here in blue. Since we had this decrease in mammary epithelial tissue, we wanted to try and figure out why. And so we started by analyzing Ki67, which is a pan-proliferative marker. And what we found is that these growing Ki67 positive cells were less frequent in LBH deficient glands. And that's quantified here. What we also found when we looked at pre-apoptotic protein cleaved caspase 3 was that there was an increase in the amount of cleaved caspase 3 positive pre-apoptotic cells. And we're actually in the process of characterizing lineage-specific proliferation and apoptosis to see whether we can attribute these effects to any particular lineage in the mammary gland. So I also did FACS analysis of those same markers that I showed earlier, and we did see that increase in the frequency or the proportion of the basal population. However, we were unable to quantify any decrease in that population when LBH was gone. What we were able to demonstrate was a decrease in the frequency of delta NP63 positive basal cells in these mammary glands. And also, when these cells were actually sorted, there was a decrease in the proportion of mammosphere-forming cells, so it indicates a loss in the frequency of stem cells. Then we got to the fun part. So we monitored these mice for a period of 18 months. That was the end point of the study. And actually a long time before that, a lot of these mice actually formed tumors. What we found is that LBH deficiency delayed tumor onset by about 9 weeks. And you may go, 9 weeks. You have to keep in mind the life cycle of a mouse versus a human being. 9 weeks, or I think about a month is about, for a mouse, about 14 years for a human being. So let me keep in mind. What was interesting is that when we did get tumors, though they came later, they were the same size, both at onset, when I actually found them, was able to feel them growing in the mammary gland, but also 3 weeks later when they were monitored, and then I sacrificed them at that 3-week point for the analysis I'll show you in a second. And this is just confirmation of LBH protein knockdown in those tumors. What was also interesting is that loss of LBH didn't have any pronounced morphological effects on the tumors, similar to what we saw in the mammary glands when we did the same thing. So you can see here, and this is that same H&E stain, that there's a lot of stromal infiltration in both the parental MMTB Wnt1 tumors and also in the LBH wild type tumors. And there seems to be less of that, but largely these LVH knockout tumors just seem to be more differentiated. You can assess that by the organization, but also the intensity of this H&E stain. Once again, we did the mass and trichrome stain as a control for the fact that we were only affecting mammary epithelial cells and did not see any difference in deposits of collagen. We did not see the same effect on growth and cell death that we saw in the mammary glands. So actually here we have is a quantification of those Ki67 and cleaved caspase-free stains, and they were largely the same. So we need to figure out what is happening. So we did some lineage marker analyses, especially considering that we knew LVH also had this lineage function. And what we found was that upon co-immunofluorescent analysis of basal keratin 5 and luminal keratin 8, we had a marked increase in the proportion of luminal keratin 8 positive cells in LVH deficient tumors. And what was also interesting is that where in the parental and the wild type control, there were multi-layer deposits of these keratin 5 positive basal cells. When LVH was lost, these cells were organizing into single layers that kind of phenocopy like a normal mammary gland. And you can see that the distribution of these luminal and basal cells actually mimics the H&E stains. You can see why we were seeing earlier some of those morphological differences that we couldn't quite characterize. And also here, you see a decrease in the frequency of delta NP63 positive basal cells that mirrors keratin 5. So you have these multi-layer deposits of basal cells in both the parental and the wild type tumors, but then a loss of those cells. Not only a change in frequency, but also a change in the organization of those basal cells. But it's important to note that though LVH is regulating lineage, it's not regulating all lineage markers. We looked at smooth muscle actin, which is used as a basal marker sometimes, but it's really more of a myoepithelial marker, meaning that there are other infiltrating cell types that will stain for smooth muscle actin. But we found that largely the distribution of smooth muscle actin positive cells was the same as the basal patterning that we had seen. The patterning of the basal markers, delta NP63 and keratin 5. We also looked at GATA3, and previous analyses had not shown GATA3 to be an LVH regulated gene. So it's not that we were necessarily expecting a change. However, the GATA3 positive cells did cluster into those islands as well, though not all of the luminal cells in those islands are GATA3 positive. The fold change in LVH is represented here, so that's the loss of LVH on the mRNA level. And we also looked at whole tumor extracts and assayed them for estrogen receptor alpha mRNA changes and found increases in the level of estrogen receptor mRNA transcripts. I haven't been able to quite get a full analysis on the protein level of that change, but that represents an interesting future direction for the work. So we had this conditional knockout, as I mentioned, keratin 14 is epithelial specific. But we wanted to know what would happen with complete LVH loss. And so we used another model that we had generated in the lab, and that was the ROSA 26 line. ROSA 26 is ubiquitous deleter line. So all of the tissues that I mentioned where LVH is expressed, the lungs and the heart and the spleen and the peripheral nervous system, LVH has gone everywhere. But it also means that it's gone in the stromal infiltrating cells in the tumor. So we have these Wnt positive ubiquitous LVH knockout mice, and we crossed them actually in this stage with a shift GFP reporter mouse. And so these mice have tagged activated stem cells that are GFP positive. Given that we knew LVH regulates stem cell activity in the normal gland, represented a unique opportunity to study the effect of LVH on cancer stem cells. And so for this part of the study, we added another gene. So these mice are both Wnt positive and SHP positive, and either have LVH or have ubiquitous loss of LVH, and that's what the knockout means. This is very preliminary because the study is still ongoing. However, at n equals 6 and 7, there is about a 40-week delay in tumor onset when LVH is ubiquitously gone. So we can see that it even further delays the formation of these spontaneous tumors. But once again, once we get these tumors, they're roughly the same size. So what that indicates is that this is actually having an even stronger effect on this tumor-initiating cell population without necessarily affecting other proportions. And as I mentioned, the stem cells in this model do express GFP. And so cryosections were taken from tumors formed in these mice, and what we found was that there was a decrease in the frequency of GFP-positive cells, which is represented visually here and quantified here. So wherever you want to look for it to make sense. But we have a decreased frequency of these cancer stem cells, these tumor-initiating cells. So our conclusions from this part are that loss of LDH decreases mammary gland hyperplasia via decreasing proliferation and increasing apoptosis in MMTV11 mammary glands, and that loss of LDH significantly delays tumor onset and changes tumor histopathology. But also that ablation of LDH leads to lineage reprogramming in favor of the luminal lineage in these MMTV11 tumors. And so now we have all these wonderful conclusions. We've summed them up into this model for now. And these models are evolving as we expand knowledge. But we have these bipotent stem cells that roughly, this is a simplified version, give rise to the basal and luminal lineages. When hyperactivation results in upregulation of LDH in these basal cells, which leads to a largely basal tumor, though these tumors are biphasic, meaning that they have a healthy representation of both basal and luminal cells. And that allows us to visualize what loss of LDH does, which is increase that proportion of luminal expansion by abrogating the expansion of that basal lineage. So what are some future directions that would be interesting for this work? I think the first one would be to assess tumors, but it would be even interesting to do mammary glands and look at FACS purified, pure luminal and basal populations from these MMTV11 tumors and see what genetic changes are being induced. A lot of this study was characterization. So we're observing a phenotype, but it's also really important to figure out by what mechanism these things are happening, because that's what allows us to actually come up with a way to stop this effect. Another interesting study, and something that I showed preliminary data for, but something that we need to expand upon, is characterizing the cancer stem cell population in these tumors, MMTV11 tumors with or without LDH. And that can be done via mammosphere assays with dissociated cells from these tumors, but also via transplantation assays. As I mentioned before, mammary adult stem cells were identified and are characterized by their ability to recapitulate an entire mammary gland from a single cell. Cancer stem cells are characterized by their ability to recapitulate a heterogeneous breast tumor from a small population. And so transplantation assays would allow us to assay whether LDH is having an effect on that particular characteristic. And also what would be interesting, I showed that levels of estrogen receptors, mRNA transcript were increased upon LDH knockdown. But it would be really interesting to do a complete protein analysis and figure out whether LDH loss is actually leading to the re-expression of estrogen receptor alpha in these tumors. Why does that matter? Well, as I keep mentioning, triple negative breast tumors lack the expression of these therapeutic targets. ER is one. And so the re-expression of estrogen receptor is being explored actually by a lot of groups as a means to resensitize aggressive breast tumors to existing and effective treatments. That's it, y'all. Acknowledgements, do some brief acknowledgements. I have to thank Dr. Regal, my mentor. She's supervised this work. We've worked together really closely over the past 3 years to make a lot of this happen. We haven't always seen eye to eye, but she bears with me, and that's all that matters. Also, Koti and Ping Ping. Koti is in the back. Ray, wave your hands. He's a postdoc in our lab. So we've gotten to work closely together over the past 1 year or two. 1 year? Yeah. And unfortunately, I'll be leaving him by himself, but you know, it's the way the cookie crumbles. Thank you for all your advice and help. in the time that you've been here. And I also wanted to acknowledge Ping Ping, who did a lot of the tumor transplantations, and then I was able to just study those tumors once they got out of the mouse, which is a big deal. I also want to thank the Department of Surgery, which was our new lab home early in my PhD. We had to get set up, and everybody was really, really helpful. I also want to acknowledge my thesis committee, Dr. Capobianco, wave your hand in the back. Yeah, that's Dr. Capobianco. I actually did a lab switch during the program. He was my chair on my last committee as well, so kudos to you for sticking with me. Dr. Alashri, who is watching via live stream, she's faculty at the University of Minnesota now. But even when she left, she continued to serve on my committee. It's been really helpful. Dr. Jorda, thank you for always being super duper positive. You're always a joy when I come meet with you. And you always bring that super needed clinical perspective to keep the work in keep the big picture visible. Dr. Landgraf, hands down the best graduate program director, like really. And the students in the room clap because we know it's true. He has a rotating door of us bringing him our problems that he probably shouldn't be in the middle of, but he listens and he's understanding and he helps as he can. If he can't help, he gives really good advice to how you can solve the problem, which is technically the point of grad school. Program director, so he's acknowledged again. Sarah and Geraldine, my program aunties, thank you guys for always being around and helping us set up for things like this, for keeping us on point with all of this annoying bureaucratic stuff. It's very confusing. And I also have to give a shout out to Dr. Bernstein, who is the graduate program director prior to Dr. Landgraf. And the OG program coordinators, Diane and Kathy. And some of y'all weren't even here for Kathy, so that's how long I've been here. Usually this is the point where people start putting up pictures of their family and of their friends, but I don't have to do that because everybody pretty much is here in this room. Mom, Dad, raise your hands. My mom is actually Dr. Bishop #1. She has an EDZ, and for as long as I can remember, has been doing work to close the achievement gap for Black and brown children in the education system. So you were my first example of what this looks like, and that was so important. Dad, when Dr. Bishop got too intense, he was always there. He's like, hey, calm it down, she's a child. And thank you for that. The little street smarts that I have, which is not much, I got from you, and I appreciate that. My grandmother, right here, raise your hand. My grandmother is a fine artist. There are a bunch of people in this world who will tell you that creatives cannot create the life that they want. And my grandma showed me that whether I decided to go the academic route or whether I decided to hit a mean jig or paint a painting, that I was going to be able to create a full, abundant life. And I really appreciate that. Every time you call me, what do you need? And often I was like, nothing, Grandma, dang. I'm sorry, I was stressed, but I really appreciate it. My Francie, raise your hand. That's my closest aunt, and as long as I can remember, she was too cool to be my aunt, so she had to come up with a special name. That's why I call her Francie. You were always there when I knew I wasn't supposed to be doing things and I was scared of my parents. Thank you, because you showed me what unconditional love without consequences looks like. All of my biomed blacks, raise your hand. Cassie, Natasha, Rhoda Wissier, Dania, Rolando, thank you for joining us. Natasha, so there's not that many of us. There's a disparity in the amount of African Americans or Black students who even pursue biomedical science degrees. And so from the time that we came in, it was kind of like, oh, I read about Xavier, sorry. Jasmine, y'all didn't raise your hand high enough. You didn't raise your hand high enough. But I've been lucky to find community among you guys, and we can hang out in and out of the lab, and that's super important. And I have to acknowledge also Dr. Dele in the back, who I met late in this journey, but she was who inspired me to get fine again, because I was letting go a little bit. She came along, and I was like, oh no, we have to go back to the gym. So thank you, because you were like, oh no sis, get it together. You cannot leave this place looking crazy. Appreciate it. So all of my friends, wave your hands. Wave, 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 wave. Candy, wave your hands. You guys are really appreciated. Even the fact that you're here on Monday at 2 p.m. is super, super important to me, and I really appreciate you. You guys have no idea what I do day to day, but you still found some encouraging words, and they really helped. And even leading up to this, there were times that I was just like, I cannot. It didn't matter what I couldn't do if I could not be social or if I could not deal with some event that was coming my way. Y'all either dragged me out the house or let me drag you out the house, which is probably more accurate. And were instrumental in making sure that I survived this thing in one piece. My last slide is a poem by the late and great Tupac Amaru Shakur, otherwise known as Tupac. It's called The Rose That Grew from Concrete. And I'm not going to read it because it's right there. If you've never read it, I suggest you get his whole poetry book because it's really good. But essentially, Tupac he was able to really make the best out of really crappy circumstances. And to my fellow grad students in the room, this can feel like really crappy circumstances. But the good thing about darkness is that eventually there's always light. And so hang in there. It gets better. And if graduate school is a bus, I've been run over about 15 times. Some of y'all are only approaching time number 3. So you have it in you, hang in there. It'll be just fine. That's really everything. I'll be happy to take any questions that you guys have now. I'm just happy I didn't cry. Let the record reflect that they don't listen, because I said don't do that. Any questions?
My baby.
Nancy.
First off, I want to say great work. For someone who doesn't understand any of this, you made it very palatable and understandable. So kudos to you. So based on all the work you've done thus far, how could this research be improved for future analysts and folks who are coming into this field?
Got it. So we actually really have a lot, and we did a lot of projects during our time together, and there were a lot of them that we just didn't necessarily get to bring to fruition. So I showed you evidence that, or I showed you that LVH is regulated by these Wnt and TGF-beta pathways, but a lot of genes don't function like in a silo. This regulation happens both ways. And we were able to generate preliminary evidence that LVH actually may regulate these pathways as well. That's really interesting because we want to know not only what happens when LVH is kind of like a driver in the process, but you know what also happens when LVH is in charge, if you will. And so I think that some of the really important work that's actually going on now is figuring out the mechanism by which LVH functions, some of its interacting partners. It's a small protein, it's intrinsically disordered, which means it has some interesting folding patterns, and it would be hard to target. What would be a lot easier to target is its interactions with other proteins. And so that'll be super important in figuring out how to move this work forward and how to get from knowing that LVH does something to be able to demonstrate that inhibition of LVH via some treatment mechanism does the same thing. Any other questions? Yes.
Hey, good job, Kilan. So you showed that loss of LVH leads to expression of ER.
Yeah.
And LVH is expressed in cancer, triple-negative breast cancer.
Yeah. So
what therapeutic implications may that have?
Right. So I talked about that a little bit. Like I said, there are other subtypes of breast cancer, more frequent subtypes. that express estrogen receptor, progesterone receptor, and also HER2, and all of those proteins can be targeted to treat those cancer subtypes. It's really triple negative breast cancer where it can still be treated via chemotherapy or radiation, but we don't really have those targeted therapies. And so the re-expression of these hormone receptors represents an opportunity to, instead of having to come up with a new treatment and go through the entire clinical trial process, we could use existing and demonstrably effective treatments and potentially like resensitize them to those. So yeah, that's definitely a long path.
So do you know how LDH is interacting in ER alpha? Like why is it allowing it to be expressed or repressed?
So LDH functions as a transcriptional activator or repressor based on its context, and it's been demonstrated that there are even other epigenetic modulations of LDH that affect function. And so there's a lot that we don't understand yet about directly how LDH is functioning. However, ER is a part of a larger transcriptional program that LDH is repressing, in the same way that delta NP63 is a part of a larger transcriptional program that LDH is promoting. And that's what I was saying, it would be really important to do those profiling experiments to figure out exactly what genes are kind of in that LDH regulated signature, so that further characterization could be done there. And then we can do some of that like mechanistic work. Any other questions? Are we done?
We hold some back for later.
I think
Yeah.
Okay, we're done.
Thank you.
I just want to make a little announcement. I want to make a little announcement. So unless things go terribly wrong, we are having a little party at 5 o'clock on the first floor, Rosenstiel conference room, which is the conference room in surgery.